Okay, this is the third lecture in molecular genetics. We are going to talk on patterns of genetic inheritance, genotype and phenotype, and one trait crosses. You see, when I write something like this, for example 1 to 4, that means there are 4 slides on genotype, and this is the first one. The next one will be number 2, 3, and 4. So this topic of genotype in 4 slides related to each other. Genotype refers to the genes of an individual, which are the unit of heredity. This is simple, and you know that. Each gene is located in a specific position or locus on a chromosome. You remember that from the definition from lecture 2, and we said along the side of the chromosome there are certain places where the gene sits, and we call it locus. The plural of it is loci. Okay, let us take the color of the eye as an example. So if an allele of one of the gene is leading to expression of blue color of the eyes, the other allele, if it is different from that one, will produce different color, say brown. Okay, so this gene is responsible for the color of the eye, so it is either, for example, blue or brown or any other color. All right, here you see 2 out of 4, so this is the second slide. Allele, of course, either dominant or recessive, and it is not necessary that the dominant one is more prevalence in the society. Okay. Of course, human beings or organisms have pair of chromosomes, two chromosomes. For example, chromosome number 2 to chromosome number 5, you have 2 chromosome number 5, one from father and one from the mother. Okay, taking the color of the skin, whether there is color, whatever color, fair, dark, black, yellow, whatever, and the thing against the colored one is uncolored or non-colored. There is no pigmentation. There is genes responsible for this. So there are 3 possibilities. Either A, this one from the father and that one from the mother, and this is homozygous dominant, and it could be homozygous recessive from mother and the father, and of course this one will be without pigmentation, this one will be pigmentation, and the third possibility is A, no matter from father or mother, it could be either of them. However, this is a pigmentation as well, because this is the dominant. If you got one letter A, capital A, the phenotype will be colored skin. So AA or small a or capital A small a. The phenotype appearance, it is the physical appearance of the trait. Okay, phenotype. So what determine the genotype? You can say it is the DNA. It is the transmit the information from one generation to other. This is DNA. However, this code in the DNA to be expressed into traits, into physical appearance, making protein synthesis and so on, you need RNA. Okay, so DNA, it is or determines the genotype. The protein, it is determine the phenotype. Once again, for skin pigmentation, the phenotypes are normal pigmentation, dominant with no pigmentation, or recessive. Let us have an example again on the appearance. This phenotype for the earlobe, you see, either it is unattached, so it is separated here, or it is attached, continuous. Is it dominant or recessive? What is dominant one? The attached or unattached? Of course, it is attached. You see, if it is capital E, small e, it is separate. Unattached, unattached. And if it is EE, capital, it is separate. The recessive one is when you got small e, small e. So these are at first row eggs and second row sperms. No, sorry. This is the eggs and these are the sperms, and this is the fertilized ova. If the sperm carry the E, capital E, and the egg E, so it is EE. This is homozygous, and it is unattached. If it is small e, small e in egg, small e in the sperm, it will be EE, and this will be attached, and it is recessive. Capital, small, again unattached, EE, because the capital E is the dominant. Okay, and I think this is simple, and you you have it for 100 times in your high school last year. Forming gametes, of course. I mean, the chromosome of human is 46. If the ovum got 46 and the sperm got 46, so that will be how much? 92. And 92 most likely to be fish, not man. So this is why, I mean, how to keep the chromosome number constant from generation to generation, you have to reduce it to half. So the egg will have half the total number in the species, and the sperm have the other half. When the fertilization occur, you will resume the number of the chromosome in that species. This is as simple as this. It is not difficult. So they are separated. Okay, 46 become 23, 46 in somatic cell, and gametes. I mean, the newer cell in the body is 46, while the gametes, sperm of the ova, 23. So this is 23 from the mother, 23 from the father. Then they will combine and return back to 46. One trait. I mean, if we are just taking, it is just theoretical, of course. We are not going to have, please let us have one trait. When we are having sperms and ova, they are producing all sorts of, but for a study, let us have just the freckles. So what happens when two parents without freckles have children? Will the children of this couple have freckles? Right, the freckles is dominant, having a freckle. So if both of them have no freckle, so it is small letters, small f, small f, small f, small f, of course they will not have it. Let us see. Yeah, this is FF. When separate, all small. You can't get capital from where? The other example is homozygous man, FF, with freckle, of course, has a children with a woman with no freckles. She is FF. Of course, these capital with another small, so all the children have all of them FF and all have freckles, of course. Yeah, you remember that? It's simple. So a one-trait cross is often referred to as a monohybrid cross because it involves only one single trait. We are talking about just a trait, just one trait, freckles. Not the color of the eye, not the color of the skin, not the color of the hair, not whether if he is tall or short, the fingers, for example. We don't have, I mean, we just forget all this and we focus on one trait for study. If we have these, they are heterozygous, FF. Each parent has two possible type of gametes. Of course, this one, for example, let us have this is the female. So the egg either have capital or small, and the sperm, you have sperm with this, and the sperm with this type, small. Okay, of gametes, and we must ensure that all type of sperm have an equal chance of fertilization. Of course, one way to do this is to have Punnett square. I will not talk about this now. We got figure of Punnett square. Now I have 11 minutes, still 4 minutes to go. Okay. As we said, it is free, segregation free, and all this will have all the possibilities. And this is 1/4 FF, 2 FF, and 1. So these recessive and those dominant, this is homogeneous and this is heterogeneous. So 1 to 1. But of course this is 3 to 1, because I mean if you go to the phenotype, these two 2s add 1, 2 plus 1, it is all of them have the dominant trait. The phenotype, as I said, yes, it's 3 to 1. Why? Because this one is dominant and this one is dominant. This one positive. 3 individual will have freckles. Yes, 3 will have freckle. One of them homogeneous and the other 2 heterogeneous. And one will not have freckles, هذا FF. This 3 to 1 phenotype ratio is always expected for a monohybrid cross when one allele is completely dominant over the other. Right, let us see how this is square, Punnett square. The Punnett square is the man Punnett is a British scientist more than 100 years ago. Okay, so if the man have capital F, small f, it's heterozygous dominant, the lady the same. So this row is for the egg. This is the egg, capital small, and here you have it capital small, and then you capital small capital, right? So all this colored, that's been 3 to 1, as we said the previous slide. Okay, this is two trait. Sorry, one. I'm sorry, we are still on one trait cross. So the parent, the previous slide they were heterozygous. Here they are homozygous. WW and small w. So for the lady, this is for the X small, and here, and you have it all hetero and all dominant. So it is all of them, all the offsprings having. I think this is very simple, and you already have it in your genetic subject last year. Another possibility or another crossing is homo, recessive, homozygous, or heterozygous, of course dominant, and here is for the egg, and here is for the sperm, and so you have 50-50. 2 of them heterozygous dominant, and 2 of them homozygous recessive. Let me just finish this slide, and or I may close it and divide it into the other section of the lecture. Thank you. Let me close it